Good afternoon or good evening, everyone. Thank you for joining us for this webinar. As a product manager for Monolith Instruments, I have the chance to attend many congresses and exchange with scientists facing the challenges of interaction with GPCR. Still recently at Drug Discovery on Target and PEX Europe, this challenge were mentioned several times. Today, GPCRs are one of the most common drug targets, even though it's one of the most challenging targets to work with, and the majority of them are even deemed undruggable. GPCR are involved in many biological processes, including sensory perception, the smell, the sight, the taste. And at the moment, it might be that you are watching at this webinar, probably drink a coffee to keep you awake. And this is highly likely that several GPCR interactions are running in the background to allow you to to have the smell of the coffee. Among the difficulties faced by scientists, measuring the binding affinities of GPCRs pose significant challenges because of limited sample availability and also the complexity of studying them in close to native state. Today, we are going to discuss how Monolith X is a well-established solution that enables you to overcome these challenges with ease by working in solution and with a small amount of sample. So, as already mentioned, GPCR are key proteins. They are cell surface receptor. They play a crucial role in transmitting signal from external environment to the inside of the cell. And GPCR are involved and are the beginning of many biological processes. And this is definitely the reason why they are a very common target for drug discovery. Today, around 34% of approved drugs are GPCR target. However, this 34% is only the visible part of the iceberg. And GPCRs remain the most common target in drug discovery, and more than 20% as function that remains unknown. And many of them are also considered as undruggable. So whether you are willing to understand better the mode of action of GPCR, or if you are searching for potential drug candidates against GPCRs, the most common challenges associated with affinity measurement of GPCR can be divided in two main categories. The first one is linked to their intrinsic complexity and their fragility. You need to make sure that you are using technologies that will preserve their structural integrity. And the second category of challenges is linked to the difficulty to produce them, and therefore they will be very precious. And you should make sure that you are using technology that will consume as low quantity as possible. Now, let's take a look at how the complexity and the preciousness of this sample can impact the measurement of the binding affinity. So first of all, the GPCR are complex, and they are complex for several reasons. It can be because they are membrane protein, but also because of their complex structure, and also because they could have different complex mode of action. And also sometimes they might require to use alternative expression strategies. So these complexities will also represent challenges to measure their interaction. So one of the first reasons of their complexity is simply the fact that they are membrane protein. They are at the interface between extracellular and intracellular medium, and it's key to reproduce a close to native environment for them. Working with membrane protein often implies to use detergent to mimic the lipidic biolayer environment of this GPCR. And unfortunately, working with detergent is usually more complex to deal with for immobilization-based technology and optical-based method. First of all, for the immobilization-based technology, it will require the use of a reference surface, and you will have to subtract the signal of this reference surface to subtract the background, or also the non-specific signal. And unfortunately, the detergent can increase the background signal, and this one might differ on the reference channel compared to the active channel, leading to some difficulties in analyzing the signal. Also, generally speaking, detergent could contaminate your instrument, and it is important while you are measuring one interaction, and if you are at the end of your interaction measurement, that you make sure that you don't have any residual detergent that are present in your instrument, and you will have to make sure that it's well clean and that the microfluidic part of your instrument is not contaminated. So that was for the part about the membrane protein. But the second thing about GPCR is that they also have a very complex structure. So they are typically structured as a 7 transmembrane helices that are connected by 3 extra and 3 intracellular loops. And when studying their interaction, it is critical to maintain their conformational stability, which will be importantly linked to their function. Once again, working with immobilization-based technology will require the need to find suitable conditions to immobilize, but not only to immobilize, also to regenerate the protein of interest on a surface. And that should be done without altering or also hindering the conformation of these helices and the loops, which are the main binding sites. The immobilization and regeneration conditions are often well optimized for secreted proteins, but the combination of hydrophobic and hydrophilic moieties of a GPCR protein make the immobilization and generation processes of GPCR more challenging. And it is sometimes very complicated to maintain their structural integrity. Another source of challenge can be their intricate mode of action and the possibility to have multiple binding sites. Whether you are willing to understand their mode of action or finding new hits, you may be looking at target agonist or antagonist, but also binding on an allosteric site. And you need to make sure that all the conformation of the protein is well preserved. Similarly, in this case, it is really crucial to measure them with minimal impact as possible on the conformation. And when using immobilization-based technology, you must ensure that your immobilization or also the regeneration procedure won't impact their native structure or their environment. One of the fourth challenge is also the fact that GPCR often require alternative expression and purification strategies. So if you think about GPCR, they are very challenging to produce, and this is one part that we'll discuss just after. And to overcome these challenges, GPCR are often sometimes studied with alternative presentation mode. That can be nanodisk, VLPs, or SMALP system. They are very common. These different systems have the great advantage to nicely mimic a membrane-like environment, but they can present some variability in composition, size, or orientation, which can then produce some surface heterogeneity when they are immobilized. So these are the part for the challenges about their complexity. Now, the complexity of the GPCR is not the only source of challenges. The other part of challenges is linked to the difficulty to produce them. That will come from a low expression level, but also the fact that most of the time you will have to purify them and also need to produce them with high reproducibility. So the main reason why GPCR are complicated to produce, and most of you are most likely well aware of that and face a lot of struggle, is because they have low expression level in native cells. And when you can expect great expression level with secreted proteins, it is definitely not the case for GPCR membrane proteins. When producing them using recombinant technique, achieving high expression level is very complicated. And the results of that is that you often have to deal with and measure interaction with minimal sample amount, and therefore they are very precious. And for sure, it is then very important to have suitable technologies that will maintain their integrity, as we discussed previously, but also that will consume minor amount of your protein. Typical ITC method, isothermal calorimetry method, have the great advantage of measuring interaction in solution, but they will consume large amount of sample. The next thing about the difficulty to produce them is that once they are expressed, we could basically study GPCR and could work directly with the membrane extract, which would be quite straightforward. This would have the additional advantage to be very close to the native condition of the GPCRs. However, the membrane extract is a very crude sample matrix, and direct affinity measurement in such matrix is very challenging. This can be for different reasons. It might be that with microfluidic-based system, it can contaminate or clog your microfluidic, and it will require some effort to keep it clean or to unclog it. And no one really like to spend this Friday afternoon to wash his instruments. Also, this extract can trigger non-specific binding signal. So if you look at membrane extract, it's quite a dirty sample. So it has a white milky aspect, and there are a lot of different proteins or different stuff in there. So it can also trigger some non-specific signal that can have a major impact on the data analysis itself. So the third thing, as most of the time it's very challenging to work with membrane extract, the logical way is to go for purification of this GPCR. But unfortunately, purification often has several drawbacks as well. So first of all, it takes a lot of time to set up the downstream of and the purification of this GPCR, but also it can lead to loss of protein of interest, making your protein even more precious. And purification process of GPCR will also typically include the use of multiple detergent and lipid that might be challenging to measure interaction for immobilization-based system. And finally, one of the consequence and one of the difficulty with the GPCR is to get a good reproducibility of your GPCR. Once you are producing one batch, and if you are producing this batch and looking at the interaction or looking at some binders, some hits against the GPCR from batch one, it might be that if you are looking at, if you are screening for several dozen of compound, it might be that at one point you are out, you don't have enough of your protein. And therefore, what you have to do is to produce a second batch. But the issue is that the reproducibility between batch one and batch two might sometimes be an issue. And therefore, it's once again very useful to use as minimal amount of sample to stay within the same batch to avoid this reproducibility issue that come from the batch-to-batch differences. So these are the main challenges about GPCR. And Monolith and its combination of spectral shift and microscale thermophoresis technology enable to overcome some of these challenges that are linked to the complexity and the preciousness of GPCR. The Monolith X will measure the binding affinity by assessing modification of fluorescence of a fluorescent probe upon binding. And the great thing about Monolith is that it will enable the measurement in solution, which will therefore allow to maintain the GPCR integrity and flexibility. Also, it will consume minimal amount of sample. Typically, you would need around 160 microliter of low nanomolar concentration of your GPCR to perform one interaction measurement. Also, spectral shift technology is compatible with all type of detergents. So no matter what are the detergents required or the lipidic environment that are necessary for your GPCR, Monolith and spectral shifts are compatible with that. And finally, it is also compatible with crude matrices. And there we have a great example to share with you after. And actually, I would like to share two different examples where Monolith instruments were used in the field of GPCR. In the first case, the Monolith X enabled searcher to find binders to the A2A adenosine receptor. So this is something we have been doing in collaboration with Celtaris. And we were looking for binders to the A2A adenosine receptor. So this GPCR is involved in many different pathways, including motor control, recognition, and neurotransmitter release. And due to its function, as you can guess, it's a very important target in drug discovery. So this A2A receptor has a low natural abundance, and its expression level might be limited, and purification still remains a challenge, as it is time-consuming and also could decrease the amount of sample you are recovering. Therefore, what we wanted to do is to get rid of this purification step and to measure the binding directly in the membrane extract. So once we had the membrane extract, we used a fluorescent tracer to target a binding site of interest. Here, that's the adenosine binding site. And we were measuring the interaction directly in the membrane extract. So the first thing we had to do was to measure the interaction between the tracer and the GPCR. And the GPCR in the membrane extract. This was done by titrating constant concentration of the tracer with increasing dilution of the membrane extract. We could see that binding of the tracer triggered an increase in the fluorescence ratio, which is the signal that we are measuring with the Monolith X and the spectral shift. And we can see that we reach a nice saturation at a dilution between 64 and 32. So once we knew that there were nice interaction between the GPCR in the membrane extract and the tracer, we then had to measure interaction between the GPCR and the different compounds. So to do that, we have been performing a competition assay where a constant concentration of the GPCR and the tracer together were titrated against increasing concentration of the compound of interest that is represented here in yellow. By doing that, we will displace the tracer and observe a decrease in the fluorescence ratio as the tracer is coming back to its unbound state. So this is how it looks like for the adenosine, which is the natural binder of this A2A receptor. And you can see here that there is a reverse shape of the curve because the tracer is going back to his unbound state. Also here, all these measurements were done directly in the membrane extract, so in the crude matrix. So this is here the example for the adenosine, but we have been running a short feasibility study with several compounds, and we were capable then to rank from weaker to stronger binder. And here also, what it allowed us is it was to perform measurement with really minimal sample amount. So only 160 microliter of the membrane extract that was diluted 40-fold was necessary. So in the end here, we had several compounds, but we could have screened for way more as we still had a lot of membrane extract. So this is for the first example. Now I would like to share another example. So in the first one, we were more speaking of an example in the drug discovery field, but of course there is still a lot that is to be uncovered about the mode of action of the GPCR. And here in this second example, I would like to share with you how the Monolith instrument, so here it was not the Monolith X, so it's still the Monolith instrument with the microscale and MST technology that was used to measure interaction of GPCR in nanodisk. So in this study that was performed by Yoshida et al., the human serotonin receptor were solubilized with detergent and purified to be presented in nanodisk. The nanodisk is a convenient way of presenting GPCR, and it will mimic their natural environment to ensure their integrity is preserved. Another great advantage of the nanodisk is the possibility to fine-tune the composition and to use different phospholipids of interest. The phospholipid composition of the nanodisk will impact on the fluidity of the membrane. And if you are looking in vivo, the lipid composition and the membrane fluidity is constantly changing due to disease or eating habits, aging, or multiple other factors. And here in this study, Yoshida et al. Wanted to study the impact of the fluidity inside the membrane on the binding of the GPCR and assess whether GPCR could still bind when the fluidity of the membrane decreases. So what they did was measuring the interaction of the human serotonin receptor, the GPCR, and they were measuring this interaction in different nanodiscs of different phospholipidic composition that had different fluidity behavior. So they were starting with the more rigid, that is the DMPC. They were also testing in POPC, DOPC, and DLPC. So they were going from the more rigid to the more fluid membrane environment. And what they were observing is that the affinity was also linked to this fluidity, and that the higher the fluidity of your membrane, the better the affinity that was observed. And the Monolith allowed them to assess the binding in solution with site-specific labeling, making use of his tag on the nanodisk. So therefore, they were sure that the impact on the structure was really minimal, as it was really in solution, and that as we were using already present his tag for the fluorescent labeling. Okay, so here we are almost at the end of the presentation. And one of the take-home message here is that Monolith and in-solution affinity measurement with low sample consumption is really a valuable tool for measuring affinities in GPCR. So it will avoid the different challenges linked to the buffer compatibility, or in this case, mostly the detergent compatibility, but will also enable you to save precious sample amount and use minimal sample and therefore you might be capable to stay within the same batch to avoid variability, or also run more potential candidates, so increase the panel of hits that you are looking for. Also, something we did not spend a lot of time on is also the time to get the results. So if you think about the interaction of the different compounds that were shown in the first study case, it took, for each interaction, it took only 3 minutes to get one single KD. So that's very super fast. And with that, I'm already at the end of this presentation. Thank you everyone for joining.